My name is Lison. I am 15. I perform a short stay at the BioOrganic mass spectrometry laboratory in Strasbourg, where I discovered mass spectrometry. Well, let's see what is performed in the lab.
Proteins ensure vital functions in all living organisms. Mass spectrometry-based proteomic analysis allows their large-scale identification and quantification. We apply proteomic analysis to characterize protein interactions and target complexes involved in mechanisms such as natural pigmentation of the skin, eyes, or retina. Proteomic analysis is also a key methodology to discover new protein biomarkers of pathologies. Murine models are not always best adapted, and more exotic models have recently proved to be very promising. For example, we do proteomics on bear models to study physical inactivity. Unlike humans, they are able to maintain muscular mass and strength despite a prolonged inactivity. We compare the proteome of active versus inactive bears to better understand the adaptation mechanisms they have developed. Another example is the study of metabolic diseases such as obesity or diabetes, for which we use a small primate model called mouse lemur, who does not develop any of these pathologies. Responses to many biological and pathologies-related questions can be addressed by studying biodiversity and trying to understand how nature has developed its own best adaptation ways.
Mass spectrometry is also widespread to study the structure and dynamics of therapeutic proteins like monoclonal antibodies that are foreseen as next-generation biotherapeutics to treat specific diseases like cancers. A particular application of mass spec is native mass spectrometry performed in near-physiological conditions. This approach is routinely used in our lab for monoclonal antibody and related products characterization. It allows determination of exact masses of the proteins or the number and distribution of drug loads on immunoconjugates. Combined to ion mobility, native MS provides information about shapes and conformations of biomolecules.